Was the genome sequence developed in Wuhan the same one being used to identify the virus? Well, the way it works is the first genome sequence, which is the paper we've been over a million times, that became the template or the blueprint. And so that's what's essentially plugged into the computer for every assembly process since then. So that first one, and I think everybody knows the paper I'm talking about, whereas the first genome that was published, that was a so-called de novo unbiased sequencing. The rest of them, once you have the quote genome, then you can use that as the template and get variations on that. So it's the first one that's the most important, and that's the one we've been investigating with Stefan and the mathematician. And interestingly, so far we've been able to find out that 98.5% of the sequences that are used, that were used to assemble that genome, based on their published data, we now know either came from human sequences, microbial sequences, bacteria or fungus, or were generated de novo by the PCR process. In other words, this original genome, which is not a sequence means you have the whole genome and you sequence the individual base pairs. What they do is assemble from little pieces an entire genome. They get a million different possibilities, and they arbitrarily pick the longest one, and that became the genome. These little sequences that they use, using their own published data, we were able to figure out, again, 98.5% of them came from somewhere we could identify that was part of that brew of the snot of the original person. Therefore, there's no possible way that could be the assembly of an actual viral genome.